Hey, I am here to try and explain DNA replication to you again, so I'm going to do my best. As with anything in biology, it's more complicated than what it seems on the surface, but keep in mind, big picture, we want to separate our two DNA strands and we want to replicate them so that we end up with two identical copies of DNA. If you look on top of the screen here, we have several different enzymes that are going to be actors in this sort of orchestrated play. And the nice thing is that for a lot of them, their name sort of has something to do with their job. DNA is directional. So what we mean by that is it's anti-parallel in its direction. So the 3-tick end means that on the other end there is a 5-tick end. So I could say that this is the 5' or the 5-tick end of the pink strand because the 3-tick end is over here. 5-tick for blue, so 3-tick strand is over on this side for the blue strand of DNA. So to begin replication, remember that we're just looking at this DNA and it looks like two little strings, but it's actually a lot more complicated than that. We have sugars and phosphates and bases, which are making up individual nucleotides. And so to make a copy of this DNA, the first thing that we have to do is we have to unwind those two strands, and then we have to separate them. And by separating them, remember when we made our models that we had those hydrogen bonds that were holding the two strands together? We have to break the hydrogen bonds so that now the two strands are separated and they are unwound and that they're not spiraled up like a double helix. So helicase is the enzyme that's going to be in charge of this. And if I could, I would make it look like a scissors because that's essentially what it does. It cuts the hydrogen bonds to separate the two strands. Where this is happening, we call this the replication fork. And think about a fork in a road is where you could choose to go this way or choose to go that way. So the replication fork is where the two strands are separating. We call this the replication bubble then, where we've already separated those two strands and we're actually doing the process of replication. When we unwind these strands, then we're actually putting more pressure on the strands upstream that we're not ready to unwind yet. And so we have an enzyme called topoisomerase, which is going to bind upstream, and it'll move down as the replication fork advances, but it's just kind of to help stabilize those strands upstream. We also have the help of some other molecules called single-stranded binded proteins. And they're going to attach to the two strands upstream from the fork. And they're also just going to help stabilize things. Specifically, they're going to help keep these strands unwound so that they don't wind up into a helix while we're trying to replicate them. All right, now we mentioned that these strands are directional. And so the way that we replicate the two strands is going to look a little bit differently. And I'm going to start with the easiest strand, which is called the leading strand. The leading strand is going to be the strand where we're starting with the 3-tick end, and the 5-tick end is where the uncoiling is happening. And that's actually really convenient because we have to build DNA from the 3-tick end on the existing strand to the 5-tick end. The new strand then will be built 5-tick to 3-tick because remember, this is anti-parallel. So to begin, DNA polymerase, that's DNA polymerase 1 and DNA polymerase 3, just think about the name. It's building DNA polymers. It's bringing in nucleotides of DNA, adenine, thymine, guanine, and cytosine. But the interesting thing about DNA polymerase, these are enzymes that they can't just all of a sudden start in the middle of nowhere and begin a new strand. They have to have something to attach to. And so that's where we have another enzyme called primase. And primase attaches to the DNA strand, and it creates a series of RNA nucleotides, which we call an RNA primer. Think about primer being like the beginning of something. So this RNA primer is a series of A's and G's and C's and U's because it's RNA, and that's going to be a starting place for my DNA polymerase. So the primase can then unattach itself. And remember, we have hydrogen bonding here between the old strand and the new strand. And DNA polymerase 3 is going to be the one that's going to come in, and it's going to bind and recognize where that RNA primer was created. and it will then begin to assemble DNA nucleotides, complementary DNA nucleotides. We call the strand that we're reading the template strand, and the new strand that we're reading is the complementary strand. So where I have an A, it's going to put a T. Move down the line, if I have a T, I'm going to put an A. Where I have a C, I'm going to put a G. Where I have a G, I'm going to have a C. And as we continue to uncoil these two old strands upstream, the DNA polymerase is just going to build one big continuous strand of DNA. And so I labeled this wrong. That's not the lagging strand. This is the leading strand. So at the end of DNA replication of the leading strand, I have one nice, long, continuous, beautiful strand of DNA, minus that RNA primer, which we'll take care of in a little bit. All right, so that was the leading strand. That's easy. The lagging strand is where it gets a little bit more tricky, because remember we said that DNA polymerase can only build DNA from 3-tick to 5-tick on the old strand, the way it's reading it, or building the new strand 5-tick to 3-tick. And so what we're going to need to do is we're going to actually build this in sections, because as the DNA unwinds, you're going to build what's in the replication bubble, and then as more DNA gets added to the replication bubble, you have to move down and add to that, and then add to that. So it's going to be a little bit more choppy. So again, we have to start with primase, and primase will attach, and it'll put in a primer, and just like we had RNA primer here, and then it'll move down and it'll put in another RNA primer. And then it'll move down and it'll put in another RNA primer. So notice that we're still having to build it from 3-tick to 5-tick on the old strand, or 5-tick to 3-tick on the new strand, however you want to look at it. And so as this replication fork moves down and the replication bubble expands, this primase will just keep going down and making new RNA primers. Remember, RNA primers are the starting places for my DNA polymerase 3. So DNA polymerase 3 will now come in, it'll find a primer, and it will attach, and it'll bring in some strands of DNA that are complementary, A's and T's and G's and C's. And then when it's done with that section, it'll come and bring in another section of DNA and add the nucleotides. And then go find another primer, and then it'll go find another primer. And at the end, this lagging strand is going to be made up of several different pieces of DNA that are not all connected to each other, and they all have their own little starting primer at the end of them. And this doesn't look so beautiful, but you get the idea. Okay, that's the lagging strand. We're almost done. Let me at least label what this looks like. So this was 3 to 5 on the old strand, which means we are building the new strand 5-tick to 3-tick, anti-parallel from each other. And here's the new strand. We're also building that one 5-tick to 3-tick. All right, so we got a good start, but we're not done because things just aren't so smooth, and we need to do some proofreading and kind of tie up some loose ends. So DNA polymerase 3 is pretty much done. And now we have the job of some of these other enzymes that we haven't used yet. And one of those, and probably one of the most important ones, is DNA polymerase 1. And it has multiple jobs, and one of the jobs that it does is it comes in and it removes the primer. And it replaces the RNA with DNA nucleotides once replication is finished, once DNA polymerase 3 is finished. So at the end, this leading strand is just one nice long continuous piece of DNA without any RNAs. It's going to do the exact same thing to the lagging strand. It's got a lot more RNA primers to remove and replace with DNA on the lagging strand, but same idea. It's moving down the line and replacing RNA with DNA. And I guess technically I could just, show that these are sections of DNA now where they used to be RNA primers. DNA polymerase 1 is also in charge of proofreading. So as it's moving down the line, if it finds that maybe you have 2 thymines attached to each other, or you have an adenine with guanine, it can do DNA excision repair and replace those incorrect nucleotides with the correct ones. And so it really makes our DNA replication much more accurate. It's not perfect. We still have mutations in the DNA, but a lot less of them because of that proofreading machinery. Then to move on, we got one more little thing that we need to do. The lagging strand has these pieces of DNA. Now we got rid of the RNA primers thanks to the DNA polymerase 1, but they are not all one continuous piece of DNA. They are fragments. And we call these fragments on the lagging strand, we call them Okazaki fragments. And we don't want to have just a bunch of little pieces of DNA. We want one nice, long, continuous strand of DNA like we had on the leading strand. And so that's going to be the job of ligase. And if you think back to signal transduction pathways, a ligand was something that bound to a receptor. It formed a bond with a receptor. Ligase is kind of like glue in that it binds different pieces of DNA together. And so here you might have a phosphate floating around that's not attached to the sugar on the next strand. And so it's going to form a phosphodiester bond between the phosphate and the sugar between those two DNA nucleotides. And it'll move down and it'll connect those Okazaki fragments together. And then it'll move back here and it'll connect those Okazaki fragments together. So even though the lagging strand takes a little bit of extra work, in the end, you do still end up with one nice long continuous piece of DNA as well. And so at the end, these two template strands will be completely separated from each other, and I will have two identical double-stranded DNA molecules. They will be anti-parallel, meaning that the 3 ticks will run opposite of the 5 ticks on the other strand. And then we would also say that this is semi-conservative. We've kept one old strand as our template, and we've made one new strand as our complementary strand. And this is just going to continue on and on and on. I mentioned in class that E. coli, their DNA polymerase machinery can do about 500 nucleotides per second. And our DNA polymerase can do about 50 nucleotides per second. So incredibly fast, but it has to be, because when you have 6 billion letters that you have to replicate every single time, you got to be pretty quick about it. So hopefully that helps you with DNA replication. I think it's easy to sit and watch somebody else do it and think, oh yeah, that makes sense. But the true test is whether or not you can get onto this and talk yourself through it and teach it to someone else.